Hello, welcome to the third part of this lecture. We will talk about G-protein coupled receptor signal transduction, and I will explain how microscopy may help us understand this interesting process. So, G-protein coupled receptors, I will give you some facts, some background information on those kind of receptors. There are about 700 genes that encode the G-protein coupled receptor in the human genome. And about 300 of those receptors are olfactory receptors, so they help to recognize odorants. Well, G-protein coupled receptors are very versatile molecules. They can recognize a wide variety of stimuli. They can recognize photons, hormones, lipids, peptides. So the very fact that you can actually see this presentation depends on G-protein coupled receptors. There are multiple crystal structures available. The number of crystal structures has actually exploded over the last decade. This has been a huge breakthrough to crystallize those kind of molecules, which was not simple because they span the plasma membrane. The receptors are characterized by very fast activation, so this ranges from seconds to even milliseconds, and this is done by a conformational change. When G-protein coupled receptors are activated, they send well-known messages within the cell, so they release calcium or cyclic AMP, which then transduce the signal. And very important for the pharmaceutical industry is that over 30% of the drugs that are currently on the market target G-protein coupled receptors, and we will discuss this later on. So in 2012, the Nobel Prize was awarded to Bob Lefkowitz and Brian Kobilka, and that was awarded for the studies of G-protein coupled receptors. So Bob Lefkowitz was among the first to identify the G-protein coupled receptor that responds to adrenaline. And then Brian Kobilka was very instrumental in solving all kinds of crystal structures. Now this is a cartoon of signaling that's activated by G-protein coupled receptors. We see here 4 different types of G-protein coupled receptors. You see they are a kind of snake that goes through the plasma membrane 7 times. And then they recognize agonists or stimuli in the extracellular space. Now these G-protein coupled receptors, as their name indicates, are coupled to so-called G-proteins. And they can be from different classes. So we have the G12 class, the GQ class, the GI class, and the GS class. And this indicates the heterotomeric G-protein that is associated with this G-protein coupled receptor. This heterotrimer, as the name says, consists of 3 subunits. And the alpha subunit is actually the G-protein. And depending on this alpha subunit, some downstream signaling is activated. So that's shown on this slide, where, for instance, this alpha 12 heterotomeric G-protein subunit activates Rho A. This can also be activated by G-alpha Q, which can also activate this enzyme phospholipase C-beta, which is responsible for the calcium increase. And on this side, we have the alpha I and alpha S subunits. They modulate cyclic AMP levels. So they control the activity of this enzyme, adenylyl cyclase, which then produces cyclic AMP. So alpha I is an inhibitory subunit and alpha S is a stimulatory subunit. So alpha I actually inhibits cyclic AMP while alpha S activates cyclic AMP levels. So you see from this slide that there is a lot of different events within a cell that can be triggered when a G-protein coupled receptor is actually activated. Now, in case you want to do drug discovery, so you want to learn something about things that activate a GPCR and maybe develop a drug that inhibits it, typically the experiments that are done in these drug discovery endeavors are adding a drug, an agonist is something that stimulates the GPCR, and then measure some kind of output. And in the drug discovery industry, calcium has been a very popular readout. And in this way, by measuring those calcium levels in cells, you can identify agonists. Now, suppose you want to develop an antagonist, so a drug that inhibits activity. What you can do is you can first add the inhibitor, then add the stimulus, and then measure the output and see to what extent this inhibitor attenuates the calcium response. This is a typical output that you can achieve in such an experiment. So on the x-axis you see different concentrations, or in this case of a compound that activates a G-protein coupled receptor, it's histamine. And on the y-axis you have some kind of output, doesn't really matter as long as you can measure something within the cells. And what you see is when you increase the concentration of histamine in this case, you see that the activity increases and you get a nice S-shaped curve, and this parameter at which you get 50% activation indicates the potency of this ligand. And in this case you see that histamine has a potency of around 10 micromolar, so 10 minus 6 molar. And you can also see from the left shift of this curve that this compound, MHP, that was also used in this experiment, has a higher potency. So 50% activation lies at a lower concentration. So you only need a lower concentration to activate the GPCR with this compound. So from these curves, you can learn a lot about the potency of drugs and their activity. Now, in this way, a lot of drugs have been developed that are currently on the market. And this slide shows a couple that may be familiar to you. So, for instance, we have Otrivin, a nasal spray that targets GPCRs, oxycodone, which is a painkiller, valsartan, that mediates blood pressure. And we also have Sirtec, which is an histamine antagonist, and this helps to treat allergies. So these are some of the well-known drugs that target G-protein coupled receptors. And as I said, these G-protein coupled receptor drugs are really a big chunk of the market of drugs. So here's a graph over time that shows how many drugs there are against GPCRs relative to all the FDA approved drugs. And you see that it's stably around 30% of the drugs targets a G-protein coupled receptor. Here's a diagram of all the diseases that can be treated with drugs against GPCRs. For instance, it can be used to treat blood or to lower blood pressure. It can be used to treat allergies. There are painkillers among the drugs against GPCRs, and also antidepressants, for instance. So there's this slide shows you there's a lot of variety in the type of drugs that you can find that target the G-protein coupled receptor. Now, this drug industry aspect of GPCR activity is of course very important. So in the end, you may want to cure a disease, and then it's interesting to find such a compound. But as I've tried to explain, it's also very interesting to learn more about what actually happens when you trigger a GPCR, and this understanding may in the end also help to design novel drugs. Now, the drug industry is screening for calcium, but of course you can also observe cells directly through the microscope and see what happens when you activate a GPCR. And that's what I want to show in this movie. So in this case we have two cells, and we have overexpressed the GPCR, so the response is kind of exaggerated. And when we add an agonist, so something that activates the GPCR, you see very stunning things in these cells. So I will run the movie. Here the agonist is added, and you see some tremendous changes in the morphology of those cells. And then at the end of the movie, an antagonist is added, and you see that the cell kind of recovers. So it's a loop of the movie. So here the cells start to relax again. Now the movie starts to play again. Here the agonist is added, and you see all those membrane changes going on in these cells. So these GPCRs, when you activate them, they have really profound effects on cell morphology. And we think this is quite interesting actually. Now, to go back to what happens when a G protein coupled receptor is activated, I have this cartoon model of activation. So again, we have here the G protein coupled receptor that spans the plasma membrane 7 times. We have agonists floating in the extracellular space. And here we have the heterotomeric G protein. Now, when we activate this heterotomeric G protein, when we activate the GPCR, the heterotomeric G protein is recruited, then it's activated, which in case of a G protein means that the GDP cofactor is exchanged for GDP, and now the alpha subunit is active. Now, to monitor this process, we make use of a toolkit that is based on FRET. And I will briefly introduce FRET to help you understand what kind of toolkit we use to measure those activities of the heterotomeric G proteins. So the classical way to measure this is by using radioactivity assays, but they have poor temporal resolution and it's also a messy method. So we are using microscopy method because it allows us to measure in real time this activation. So as I said, for this we will use FRET, the FRET technology. We will use 2 fluorescent proteins. In this case, it's a cyan fluorescent protein and a yellow fluorescent protein. And what happens if there's no FRET? When we excite the cyan fluorescent protein, we get cyan fluorescence. However, if you bring those 2 proteins close together, as is shown in this lower panel, what happens is that the cyan fluorescent protein can transfer the energy to the yellow fluorescent protein, which then starts to emit light. So this cartoon shows that if we bring those fluorophores close together, we get, instead of only cyan fluorescence, we also get yellow fluorescence. And by monitoring these 2 signals, we can study whether proteins are close together or not. This is a cartoon model of spectra, and they illustrate what happens when FRET occurs. So what you see here are the CFP and YFP molecules and their distance. And if you bring them close together, what you see in the spectra is that this peak starts to collapse, while the other peak rises. Now this peak is the cyan signal that collapses when there is FRET, and on the other hand we have this yellow signal that increases when FRET occurs. And this is the readout that we will use to measure activity of the G proteins. So it's again explained on the next slide, again with the spectra. This on the left is a spectrum where we have high FRET, and on the right we see a spectrum where we have low FRET. Now, what we have done is we have this heterotomeric G protein complex that consists of 3 proteins. We have tagged the G alpha subunit, so one of the 3 subunits with a cyan fluorescent protein, and the other 2 subunits, the beta and the gamma subunit, with a yellow fluorescent protein. Now, if this protein complex is in a resting cell, it's not active, and the complex is associated. Now what happens when we activate the heterothermic G protein? The alpha subunit binds GTP and the protein complex dissociates. And now the distance between the two fluorophores becomes bigger and we get a loss of Fred. And this is something we can measure. So initially we have high Fred. After this dissociation we have low Fred. And in case of high Fred we have a lot of the yellow signal, and in the case of the low Fred we have more of the cyan signal. And this is what we can measure with our microscopes. And the nice thing is that we can actually take images of these two channels, the cyan channel and the yellow channel, and then we can make an image that displays the ratio of the two signals. And that is shown here below. So we have the cyan fluorescent protein channel, and this is a cell that shows the distribution of this intensity. And there's another image of exactly the same cell, but now imaged for the yellow intensity. And if we make a ratio of those two signals, we can actually see within the cell where the activity is high, so where we see those white pixels, and where the activity is lower, for instance, within this cell. So with this technology, we can not only monitor the activity, but also where this activity is high in a cell and where it is low. This is an example of such a Fred experiment over time. In this case, it's another probe that responds to calcium. Here we have the cyan signal, here we have the yellow signal, and in this case, when calcium levels rise, we get an increase in Fred, which means a decrease in CFP and an increase in YFP. And you see that in the both channels. And again, on the right here, we see a ratio image. And this ratio image now reflects what happens with calcium. So this is a very nice technique to measure in real time in cells the concentration of calcium, but you can also measure, for instance, the activity of GPCRs and heterothermic G proteins. So it's a very powerful technology. So again, to remind you how this system works, we have initially the complex with high Fred, and after the GPCR is activated, we get a decrease in Fred. And the data then looks like this. On the left you see this nice cyan trace and the yellow trace. In a resting cell we are right here, and then when we activate the GPCR in cells, we get a loss of Fred. So we get an increase in the cyan fluorescence and a decrease in the yellow fluorescence. And at some point, right here, we add an antagonist, so the GPCR is inactivated, and we see that the cyan intensity goes back to baseline and the yellow as well. And typically these data are plotted as a ratio, so you ratio the CFP versus YFP, and you get these kind of curves, which nicely show the activation as a loss, and then if we inhibit the GPCR, it comes back to baseline. And this is now a way to measure GPCR activity over time. So to summarize this part of the talk, I've shown you that G protein coupled receptor activation not only releases calcium in cells, which is nice if you want to do drug discovery, but we see also profound changes in cell morphology. So there's really something going on with this cellular morphology if you activate a G protein coupled receptor. And with Fred technology, we can actually measure this G protein activity, and we can measure it within cells. We can see where the activation occurs and what the timing is. And now with this nice technology, we can learn more about how this GPCR activation affects cellular morphology. And this will be the topic for the next part of this talk. before I explain how we study how G protein coupled receptor activation affects cellular morphology, I want to introduce you to this nice biological system. And it deals with blood vessels and the cells that line those blood vessels. This topic is also heavily studied by Jaap van Buul, who will give a lecture also in this series of clinical cell biology. Now here we have a blood vessel, and the inside of the blood vessel is lined with specific cells which are endothelial cells, which you see over here. They play an important role as a barrier between the blood and the surrounding tissue. Even so, it's a stable barrier, still white blood cells can permeate this barrier, which is interesting by itself, and it will also be dealt with by Jaap in his lecture. This is a movie of what's going on during this process of transendothelial migration, and it's just to show you how dynamic this system is. So what you see here is a cover slip with endothelial cells, and they are kind of difficult to see. At some point we flush in these white blood cells, these little balls, and they start to crawl on top of the endothelium, and at some point move under these cells. So these endothelial cells, they are quite flexible. They can have these white blood cells permeate them, and they can rearrange their cellular structure according to this phenomenon. Now the endothelial barrier is not only affected by these white blood cells, it's also its structure is also heavily affected by G protein coupled receptors. So G protein coupled receptor signaling really can change the barrier strength of this endothelial cell layer. And in case of misregulation, this may lead to edema and sepsis. So what can happen if the endothelial barrier is activated under disease conditions is that there will be holes in this endothelial barrier, and the barrier function is no longer intact, and this leads to all kinds of problems. Disruption, for instance, can be triggered by these molecules, thrombin, histamine, platelet activating factor, which all activate G protein coupled receptors. But on the other hand, there are also reagents that activate or strengthen the barrier. And one of those is sphingosine 1-phosphate. And the question really is, how do these molecules that all activate G protein coupled receptors have such a different effect on endothelial cell morphology? And this is something that we are very interested in. Now, this is an example of two endothelial cells, and they're activated with thrombin, from which we know that it can cause holes in the endothelial barrier. And what you also see when the thrombin is added, you see a change in this color. I will explain later what that means. But what you also see is that the cells start to shrink, and this of course then leads to these holes in the barrier. Now these cells were transfected with a probe for a certain G protein, in this case it's a G13 class, and we were interested in measure its activity. And the activity is shown in one of those typical curves over here. So here we measure the activity of the G protein over time. This is the baseline where no stimulus is applied. And then when we activate it with thrombin, we see an activation of this G protein, which then returns to some lower level. And you see that it nicely is plotted over time. So this tells us that thrombin activates this heterotomer G protein. And this is already well known, and it's also known that this affects cell shape and that the cells start to shrink. Now we were interested in this compound, sphingosine 1-phosphate, because it strengthens the barrier, so it has the opposite effect as thrombin. And the question was really, what kind of heteromeric G proteins are activated by sphingosine 1-phosphate? And this was kind of a complicated question because there are multiple GPCRs that can bind sphingosine 1-phosphate and trigger downstream signaling. So here again the question, which heterotemer G protein families are activated by sphingosine 1-phosphate receptors? So first we took our probes that can measure all the heterotemer G protein activities and checked whether they were activated by sphingosine 1-phosphate. So you get these typical curves with baselines, we add something and then we look at the reaction. Now in this left graph we see the response of the G-alpha I sensor, which clearly responds to sphingosine 1-phosphate. Here we have a G-alpha 13 sensor that also responds. We have here a G-alpha Q sensor which does not respond to sphingosine 1-phosphate, and we used a control, a positive control, histamine, that shows that this sensor is actually, it can be activated in endothelial cells. So it's not a general problem with this sensor. This sensor operates as it should, but it just doesn't respond to sphingosine 1-phosphate. So what we've learned from these experiments is actually that we have G-alpha I and G-alpha 13 heterotemer G proteins that can be activated by sphingosine 1-phosphate. Okay. The next question is, what happens when you activate those heterotemer G proteins? What kind of effect do they have on cell shape? And for this, we used a specific method that's a label-free method that doesn't use any fluorescent probes. And it has some advantages. And probably you can come up with a few advantages once you know what this technique does. Now the way that it works is that we have a current flowing through our cover slip. So when we have a cell layer here, it works as an insulator. So the current that flows through this cell layer is lower than what you would have if there is no cell layer. So if this cell layer is intact, we have some resistance. And the resistance we can measure over time. And a drop in resistance basically means that these cells start to contract and generate holes. Now, if the barrier is strengthened, so you get a better overlap of the cells, then the resistance increases. And this technology is as abbreviated as ESIS. What you should remember is that this is a population analysis. So we have a lot of cells on this monolayer, and then we kind of measure how this whole monolayer reacts. Now this shows you a graph of the resistance over time, and we measure this for two agonists, so two molecules that we use to stimulate this monolayer of endothelial cells. One is thrombin, for which we know that it generates holes, and we see that, right? We see a drop in resistance when we activate thrombin. If we repeat this experiment for sphingosine 1-phosphate, we see an increase in resistance, and this fits with the idea that sphingosine 1-phosphate increases barrier strength. Now we know, and we have seen, that thrombin activates G-alpha 13. On the other hand, our G protein probes have shown that sphingosine 1-phosphate activates G-alpha I and G-alpha 13. So the question, what is going on here? It looks like this G-alpha I has some kind of opposite effect to G-alpha 13. And another interesting aspect is that we know that G-alpha I and G-alpha 13 are activated simultaneously. So what happens in the cells when these two heterotemer G proteins are activated simultaneously? Apparently, the net result is an increase in barrier strength. And so the question is, can we understand that? So we started to look at individual cells. This is again an endothelial cell that is activated with sphingosine 1-phosphate. And I'll run the movie, and what you see over time, hopefully, when you look at the shape of the cells, I'll run it again. Sorry. What you see if you look at the shape of the cells, it starts to expand. And of course, this fits with the idea that sphingosine 1-phosphate increases barrier strength, so there's a bigger overlap because the cells start to expand. We can quantify this over time. In this graph, you see the area of the cell. So this is the area of a resting cell. When we add sphingosine 1-phosphate, the area of the cell starts to increase. We've also indicated that with this color map, in which we overlap the cells before and after activation. And if you see blue, this is the area where the cell expanded, and if you see red, it's the area where the cell contracted. And it's clear that overall this cell has expanded. So to summarize this part of the talk, sphingosine 1-phosphate activates both G-alpha I and G-alpha 30, and the net result of these activities is actually increase in barrier strength and an increase in the area of the cell. Now, what is going on would be the next question. So we know from previous work that this alpha 13 activity, that is for instance triggered by thrombin, is mainly associated with an area decrease. The cells shrink. Now that would probably mean that if we see a net increase, that this alpha I is responsible for an area increase. And there is a very nice tool that we can use, which is pertussis toxin, which can inhibit the G-alpha I protein. So for sphingosine 1-phosphate, we know that both alpha I and alpha 13 operate at the same time. What would happen if we add sphingosine 1-phosphate to cells that have been treated with pertussis toxin, so in which the alpha I activity is completely blocked? Now here you see the result of the measurement of the G-alpha I activity. It's kind of a control experiment that depicts that if we normally add sphingosine 1-phosphate, we get activity of G-alpha I, but the treatment with pertussis toxin completely blocks the G-alpha I response, as we would expect from this inhibitor. It shows that it is a nice selective inhibitor. Now if we look at the cells, and that's what we did, we get movies like this. So again, we measure here the response of a FRET sensor, which is right now not that interesting. What you see now when you add sphingosine 1-phosphate to this cell that is pre-treated with pertussis toxin, that instead of an area increase, we get an area decrease. It's clear from the movie, and it's also clear from this analysis, in which we see now a lot of red area, which is the area that has been contracted. So while sphingosine 1-phosphate normally increases area, it now decreases. So it starts to look more like the thrombin response. We also did the same experiment with this ESIS methodology, so the label-free methodology. Here is the baseline. So normally sphingosine 1-phosphate causes an increase in the resistance, but when we pre-treat the cells with this pertussis toxin, we get a decrease in the resistance. So indeed, it will start to look like a thrombin effect. So to summarize this is a model of what we think is going on, that normally we have alpha 13 activity that activates a certain rho GDPase, and that then causes contraction. And we have another pathway that does exactly the opposite. So this pathway activates G-alpha I, activates also a rho GDPase, which is responsible for cell spreading. And in case of sphingosine 1-phosphate, both these pathways operate at the same time. The net result is spreading, but we also have a little bit of this going on. But the net balance, because this pathway is activated stronger, is cell spreading. And of course, it's pretty curious that this cell has both these opposite pathways going on. And of course, the experiment with pertussis toxin nicely demonstrates that if we knock out or inhibit this G-alpha I signaling point, the net result is contraction. So by tweaking this G-alpha I activity, we go from spreading to a contraction. And this then leads us to these conclusions. We have shown that sphingosine 1-phosphate activates both G-alpha I and G-alpha 13. And now this has important implications for GPCR control of barrier function. So if we want to prevent barrier breakdown, we have several options. We can either inhibit the G-alpha 13 activity, or we have to find ways to activate G-alpha I activity. So there are basically two components that we can start to tweak. And of course, there's also opportunities when we identify all the downstream effectors, so the road GTPases and the components that activate the road GTPases, to tune those and to further be able to control this barrier activity. So that was the end of this third part, in which we have looked at how G-protein coupled receptor activates cell morphology. And I would say this also illustrates how GPCRs act on a very physiological relevant process, namely the intactness of blood vessels.
Well, that's the end of the film. Now here's the meaning of life. Thank you, Brigitte. Well, it's nothing very special. Try and be nice to people, avoid eating fat, read a good book every now and then, get some walking in, and try and live together in peace and harmony with people of all creeds and nations.